Today, we are diving into a really interesting and surprisingly simple topic: glycogenolysis. It is one of the key pathways in carbohydrate metabolism. Let's start with a little overview of glycogenolysis to get things going. Glycogenolysis is simply a part of glycogen metabolism. More specifically, it is the breakdown of stored glycogen into glucose for energy. It is the opposite process of glycogenesis. Glycogenolysis occurs in both the liver and skeletal muscle, where glycogen is stored. It happens when the body is in a fasting state, when blood glucose levels are very low, or during the fight or flight response. The purpose of glycogenolysis is to produce glucose from glycogen and provide fuel for the body. As we know, the liver is the primary storage site of glucose, where it is stored in the form of glycogen. During the fasting state, or periods of low blood glucose levels, the pancreas sends a signal to the liver in the form of the hormone glucagon to release glucose. Similarly, during the fight or flight response, the adrenal glands send a signal to the liver or muscle in the form of the hormone epinephrine to release glucose. Either glucagon or epinephrine triggers the process of glycogenolysis in the liver and muscle. These hormones bind to receptors on the surface of these tissues, which then activate a G protein. The G protein, in turn, activates adenylyl cyclase, which causes an increase in cyclic AMP. Keeping these in mind, let's move forward. Cyclic AMP activates protein kinase A, or pKa, which converts inactive phosphorylase kinase into active phosphorylase kinase. The active phosphorylase kinase then phosphorylates and converts inactive glycogen phosphorylase into its active form. Finally, active glycogen phosphorylase acts as the key enzyme that breaks the glycosidic bonds in glycogen to produce glucose 1-phosphate. Again, keeping these in mind, we finally move to how glycogen is converted into glucose for energy, which occurs through a series of very simple steps. First, the breakdown of glycosidic bonds in glycogen begins at the outer ends of the branch molecule. Glycogen is then converted into glucose 1-phosphate by the enzyme glycogen phosphorylase. Glucose 1-phosphate is then converted into glucose 6-phosphate by phosphoglucosamidase. This is an isomerization reaction, and it is reversible. Finally, glucose 6-phosphate is converted into free glucose by glucose 6-phosphatase, and released into the bloodstream to fuel the brain or other tissues as needed. However, this final step occurs only in the liver. In muscle, glucose 6-phosphatase is not present. As a result, glucose 6-phosphate enters glycolysis directly, producing ATP for immediate energy during muscle contraction. To wrap this up, I've divided glycogenolysis into 3 simple parts, and showing them step by step. It really helped me remember the process easily. I hope this breakdown helps you too. With this, it is the end of this video. Thank you so much for watching, and stay tuned for infographics on glycogenolysis on my website. Click link on my bio. Please like and share the video and subscribe my channel Flipping Nutrition if you like the content.